time to be with you to present my latest work, which had been done in Kuwait Institute for Scientific Research. The work had been done for the first time in Kuwait for detecting the tomato yellow leaf curl virus, a special type of gene that nobody detected before. And the full genome of the virus was submitted to the gene bank. Like this.
This one forward.
I'll give you a small introduction about the problem when it was started. A few years ago, the disaster was started when we lost most of the tomato crops, which was planted in the greenhouse. Also the field one. I have plenty of pictures to show you. The major responsibility of these diseases and loss was the virus, because this virus is a transplant to the plant by the vector, which is whitefly. During the survey, which was done in 2013, we collected some samples and which were observed the symptoms of this virus. The yellow leaf curl, upward cupping, curling, and dwarfism. The first step of this successful management of the controlling this virus is to identify the problem. The main objective of this project was to adapt and optimize a rapid and clear characterization molecular test, which will depend on the PCR. Two methods we used in collecting the samples from North Kuwait, which was Abdali, and South Kuwait, which was Wafra. We have two district areas for agriculture, and we are, if you can see in the north, we are neighbors to Iraq and Iran, and in the south we are very close to Saudi Arabia. The collection was done for 2 periods of time, because the tomato plants grown into seasons. One season in the winter, which is done in the greenhouse, and the second one, which is in the previously the summer months, it's done in the greenhouses. This is the group of our work, which they are sampling here. Mr. Hamid is sampling the white flies, and here he collects also the fruits. There I'm collecting from the implant which was done in the control room, and here is the greenhouse. The detection of TYLCV was done by doing the DNA extraction. Simply follow up the De La Porta method. And then the PCR amplification, we use 2 pairs of primers. The rolling circle amplification done to complete the partial sequenced genome, while we done the enzyme digestions with 10 types of enzymes. The restriction enzyme was run, and the product was run on the agarose gel. After that, the fragment of 2,800 was purified and kept for another reason, which we then cloned it and kept it for diversity study. After the sequencing, we done a blast with the gene bank sequences which were present there. We found that our detected gene of this type of tomato yellow leaf curl virus was nearly And the result I will show you was nearly similar to the Iranian one, the Shiraz. Here the symptomatology, which we were find, it was a highly incidence of loss, 90% about the crop we lost it. Viral symptoms were varied. I'll show you by pictures better. Here the stunt in the growth, you can see clearly how is this the healthy one and this is the diseased one. And you can see the leaves, how it is upward curling. The cupping, upward leaves, this is a symptom of the TYLCV. Also the leaf deformation. By the way, the field also plants. We done the detection with both plants. Leaf curling. All the pictures show very clear. Here, leaf yellowing. It started whenever the whitefly transfer the virus. It doesn't matter which age of the plant, whether it's started planted or in the pre-flowering, after flowering, or in the fruiting. Here you can see it is in the fruiting time. You can see the high population of the whitefly. With this presence, you can extremely say it is transferring the disease. And here we found a very rare symptoms that nobody yet identified in the world, which is purpling of the leaves. I think Florida, they work on it, but they didn't know that it is virus or fungi or whatever. We are sharing our work with them to give them samples to know what's this problem. Sorry. You can see the problem. It's happened and destroyed all over the plants. Here's the gel shows the positive gene. By using the PCR, as I said, we done the full-length genome, and those two primers works with us. And the samples were from Wafra place, which is south of Kuwait. The gene bank submission was done, and then we got the accession number. The amplicon was shown a new species of TYLCV with a similarity of 90% only with Shiraz strain. In general, in our conclusion, this study will ensure that the presence of the TYLCV in Kuwait, it's there. So how can we do solve this problem? We are working now with our group to make another brochure. We done one brochure for our farmers. You know, most of the farmers, they are not educated. They don't know how to use the chemical insecticides to get rid of the insects, or to put the sticky mats, or to close, to use the double door greenhouses. So we gave them many advices to use, and once the disease started, the problem started, they have to take off all the plants away and burn it up. Otherwise, there is no solution for this virus. So the early detections help us to overcome this problem. And now we are optimized with the procedures that we use, whether the PCR procedure or the by eye. We trained our staff in USA, in Florida University, and I done my work in Wisconsin-Madison University and Washington State University. So we are in share with those three universities to ensure our work and to certify our full genome sequence. Also, in the future, we want to do the certified seeds for tomato. Not only import them, we have to do the certified one, which is belong to our virus. So the clones that we kept, we can use it later on this matter. I would like to thank you, and sorry for my voice because I'm sick. Thank you. Excuse me. Yeah.
I'm just wondering, how does the white fly transmit the virus? Does it eat the leaves or does it
As you know, the white fly is a sucking insect. You don't know? Okay. It's one of the sucking insects. So once it's dying, or it acts like a capillary tube. Once it comes to the leaf and suck the sap, the virus will come to directly transferred to the leaf directly by by the systematically transfer. The leaves will be sick, or symptoms will appear after 6 weeks. Otherwise, we can do one thing, because we are collecting the white fly, we can know that it's a diseased one or not. It's carrying a disease or not. Because of that, we done the early detection. So maybe the white fly, it's not carrying a virus. It's healthy white fly. But it will affect the plant, because by sucking the sap of the leaf, so it will, and the white fly always live beneath the leaf, not on the top. If you want to search for it, just overlap the leaf, you will find it. And special type of months, special time of months in Kuwait, when we have a little bit of humid, you can find those white flies. Am I answering your question?
Absolutely.
Thank you.
So I have a question then.
Yeah.
So what is the most efficient insecticide for whitefly?
We use bioinsecticide. We done it locally in Kuwait, but we used to brought it from USA, Biactol. It was acting very nicely. 1999. Yeah, it's safer, but in Kuwait, if they know that it is bacteria, they will not use it. They prefer chemicals. Yeah.
Well, but it's more green and safe type of
Yeah.
insecticide. But another way of doing it, of course, genetically modified.
We have to, but
it's long-term project and probably not as easy.
yeah.
And
it's not allowed, doctor. In Kuwait,
yes.
They are not allowing genetically modified food. That's why we didn't go on this.
All right.
Thank you.